Before we begin our discussion of fungi, let's remind ourselves of how organisms are arranged on planet Earth. We have been studying the cellular design of organisms. These microbiological concepts we have covered are common to all life on Earth. Our big punchline when transitioning from microbiology to macrobiology is genotype gives rise to phenotype. Evolution is a look at how phenotypes have survived or not survived based on the environmental conditions in the geographic region in which the organisms live or have lived. For example, we know that 3 times in geologic history of Colorado there was great inland sea. Therefore, our geology, the actual sediments, are remnants of sea bottoms where organisms were preserved in the sands and pushed underground and thrust back up. These fossils tell us about the organisms that once lived in Colorado. This kind of fossil evidence tells a story about organisms that leave impressions or their actual bodies in the sediments that are preserved over time. What about soft body organisms like bacteria and archaea? Fossils are not very helpful there. Therefore, scientists have made assumptions that turned out to be wrong. In the 20th century, our technology gains greatly increased our understanding of how organisms function and how they are related. Microscopy and genetics have changed the science of biology. This is why biology is the science of the new millennium. The classification of fungi is a great example of a more recent biological understanding. We used to classify fungi as plants, but now we understand that their metabolic processes are more like animals. The current model for organismal relatedness is depicted here. Remember that this diagram is a model. The model is informed by our current understanding of the genetic relatedness of organisms through evolutionary time. We know all about genetic relatedness now that we have explored the microbiological side of life. As we pivot towards macrobiology, we focus on the relatedness of species. We also focus on form and function. We are now investigating the question, why do some phenotypes facilitate fitness such that these organisms persist on planet Earth through evolutionary time? Recall from our discussion of evolution that fitness refers to an organism's ability to survive to reproductive age, find a mate, and produce offspring. This is the point of it all, the reason biologists argue organisms have a fight for life. The survival of the fittest and descent with modification concepts proposed by Darwin and supported by decades of evolutionary research are based on the assumption that there is a reason to wake up in the morning and survive. All fungi are absorptive heterotrophs. They are other feeders that absorb their nutrients from their environment. There are three types: saprotrophs, parasites, and symbionts. Saprotrophs feed on dead and decaying material. This ecosystem service is central to decomposition on Earth. Parasitic fungi live on or in another organism to the detriment of the host species. Fungi can live in diverse habitats. Most are multicellular, although some are unicellular, most notably yeasts. Fungi are non-motile. In the broader scheme of classification of life on Earth, fungi are in the domain Eukarya. Remember that eukaryotes have membrane-bound organelles and DNA secured in the nucleus. Symbionts work together to survive with each mutualistic partner benefiting. For example, lichen is a symbiotic association between fungi and algae. So within the kingdom fungi, there are several phyla. But before we get there, remember our classification scheme. Phylum is an intermediate classification. Remember Dumb King Philip came over for great spaghetti. For a long time, fungi and plants were classified together. Now they are in different kingdoms due to their metabolic differences. Their metabolic process explains why they play such a crucial role in decomposition and nutrient cycling on planet Earth. Through enzymatic release, fungi digest food outside of the cell body and then absorb them through the cell wall. In this process of digestion, Fungi also make nutrients available to their mutualistic partners. For example, in addition to lichen, symbiotic relationships exist between fungi and plants in soil. Soil fungi, mycorrhizae, and actinorhizal grow into the plant cell membrane where they exchange nutrients derived from the soil in exchange for carbon-based photosynthetic products. The fungal hyphae vastly expand root area. Modern studies indicate that this fungal relationship is more important to plant growth success than we had previously concluded. For example, paleobotanists are studying the fossil evidence of arbuscular mycorrhiza to characterize the history of plant-fungal symbiosis through geologic time. The fungal-plant relationship is one we will explore a bit more in the plants unit. The fungi are entirely composed of hyphae, which are the anchoring components. Collections of hyphae are called mycelia. They create a fruiting body of fungi. Septate hyphae have cross walls between the cell walls, and non-septate hyphae have no cross walls. Non-septate hyphae are a kind of continuous structure with floating nuclei. Both are composed of chitin, the kind of carbohydrate or complex sugar. Chitins are also found in the exoskeletons of arthropods. Remember that fungi are absorptive heterotrophs. They secrete digestive enzymes into the environment. These enzymes break down nutrients into absorbable forms. This all happens outside of the chitin wall. This should tell us that the chitin wall is a restrictive barrier, as all cell membranes are. Fungi have sporangia that can be formed in three ways. Spores can develop from terminal cells of reproductive hyphae. They can develop in sporangia, which are enlarged structures at the end of specialized hyphae, or in fruiting bodies that contain spore-forming hyphae. The spore is a unit of reproduction, a gamete, found in fungi, non-flowering plants, bacteria, and algae. In asexual reproduction, hyphae produce mitospores by mitosis. Notice the word similarity. Notice that we're talking about haploid cells that are just reproducing clonally by mitosis. The evolutionary significance here is that asexual reproduction results in no genetic mixing of information, so the offspring are identical to the parent cell unless some kind of mutation occurs. Arbuscular mycorrhiza are an example of asexually reproducing fungi. In zygotic sporulative reproduction, two haploid mating types of hyphae called gametes can exchange genetic information when in contact. This process is called conjugation. Fused gametic cells are called zygotes, which make them myospores through meiosis. Myospores germinate to make new haploid mycelia. This is often denoted by a plus and a minus, which you can think of in your mind as a male and female, but they're not different in terms of their reproductive structures in any way. They're just two different mating types. They can fuse together, but as soon as the diploid zygote occurs, meiosis ensues to go back to a haploid stage. As a result, the majority of the fungal life cycle is in a haploid stage with a very brief diploid stage. Now, let's take a look at some different phyla of fungi. In the phylum Zygomycota, we have bread mold. There are three kinds of hyphae. Remember, hyphae are just really fine filaments of cells. These fungi have stolons, which are above ground body parts that connect organisms. For example, strawberries and spider plants have stolons. They can send off little baby plants, and then when the baby sits on the soil, it roots. Over time, the stolon decomposes, and the baby is an independent organism. We already talked about rhizoids. Rhizoids are root-like structures. Their primary function is anchoring. They don't do all the jobs that real roots do. In fact, fungi don't have any real roots. Finally, we have sporangia. The sporangia are a specialized fruiting body. Some fungi exhibit both asexual and sexual reproduction. Asexual reproduction results from conjugation when there is a physical connection between the hyphae of two mating types. We'll go through this process in more detail in the next couple of slides, but essentially the zygote, the diploid, can go on to produce the whole organism, but not before meiosis occurs to reinstate the haploid condition. Now let's look at the Ascomycota. These are the sac fungi. They exhibit sexual reproduction. We're going to talk about two processes here repetitively, so hopefully it will become clear what we're talking about. In plasmogamy, we have the fusion of haploid gametes to make dikaryotic hyphae by mitosis. In plasmogamy, we have cells becoming one, but the nuclei are still independent, and that is why we still consider this haploid. In karyogamy, we will be hyphae with two nuclei. In the fruiting body, the ascocarp, the ascus develops, and then we see the process of karyogamy, where two nuclei actually fuse together to make a diploid zygote, a nucleus with two copies of chromosomal information. Then immediately following that, meiosis ensues to reinstate the haploid condition. Then the haploid nuclei are replicated through mitosis to produce ascospores. Then we have dispersal, and the whole process starts again. Some common sac fungi include Dutch elm disease, in which beetles actually attack the trees in the carrier fungus, which results in secondary attack on the tree. We also have the delicacy truffles and yeast in the Ascomycota. Lichen are a symbiotic relationship between fungus and alga, and this usually involves a sac fungus as the fungal partner. The fungus absorbs water and nutrients and anchors the organism, while the algae conducts photosynthesis. The algae delivers carbon-based compounds into the organism. There are three types of lichen: crustose, foliose, and fruticose. I think I've seen all three in the Colorado forest. Typically, you see these in the subalpine life zone where it is pretty moist because the fungi are non-vascular. They cannot transport water. They can only absorb moisture from the nearby environment. The club fungi are the Basidiomycota. These exhibit sexual reproduction, which produces basidiospores. Notice that we see this word carp again as we discuss this Basidiomycota. The suffix carp should tell you fruiting body. Again, we have plasmogamy occurring where we have the fusion of haploid gametes to make a dikaryotic hyphae by mitosis. Then karyogamy occurs where the nuclei fuse. So now there is two sets of genetic information. This is a diploid, which then undergoes meiosis to produce haploid basidiospores again. Notice the major difference between basidiomycetes and ascomycetes is that meiosis is not immediately followed by mitosis. Some common club fungi include rusts, which infect plants and can cause a lot of agricultural damage. We also have shelf fungi and then culinary mushrooms. We discussed the Glomeromycota earlier when looking at soil fungus plant associations. They produce asexually through conjugation. The glomeromycota are a fairly recent classification. In general, fungal classification is a work in progress. As more genetic research is done, more is known about the genetic relatedness of species. I hope this video gave you a thorough introduction to fungi, and in the next video we will talk about our lab procedures.